Hello, guys. So, how are you all? So it's been a long time. I hope you have thoroughly enjoyed your Diwali holidays and all, right? So, how was your festival, guys? I hope everything went good and everyone had a safe Diwali, right? Good evening, Osama Sheikh. Good evening, Hasnain Raza. Good evening, Priyanshi. Good evening, Dr. Benedict. Hello. Right. Fine and shine, everything beautiful. That's great. Hello, Priyanshi. Good afternoon. Vidoc site. Good afternoon. Medical Love Abhay Sonwani. Good evening. Hasnain Raza, yes, even I enjoyed, but I was really busy with preparing for these lectures and all. KK, yes. Come on, let's wait for 2 more minutes and then let's start. When we get sufficient amount of students, we shall start, guys. Okay. Rishi Raj, namaste. Vishnu Naik, hello. Agya Savlani. Anatomy session will be on 1st and 2nd of November. Okay, 1st and 2nd of November, we shall be discussing many more interesting topics on anatomy. But as far as today and tomorrow, we shall be discussing the microbiology, as you also asked me to teach microbiology, right? Rishi Raj, thank you so much. Tino Tenda, thank you. It's an honor. Thank you. Roopshika Datta, good evening. Right, guys. I hope you am I audible to everyone, guys? Am I audible to everyone? Yeah. Fine, that's great now. So guys, previously we have discussed regarding the catalase, right? We have discussed regarding the catalase organisms. I hope all of you have gone through that. Rupshika, don't worry. Don't worry, I'll help you, right? So micro, I know micro is a little bit a dry topic, okay, but I'll give mnemonics and I'll teach you as easy as possible. But again, I'm telling you, everything you have to learn with a concept, okay? Either it is micro or NAT or anything, there is a reason for everything. So you have to learn with the concept, only then the concept stick to your mind. And don't worry, I'm here to help you, okay? So attend this session and please let me know your feedback, okay? Video is lagging. మళ్ళీ చెక్కింగా లేకపోతే ఫైనల్ అది? So guys, look here. Previously, we have discussed regarding the catalase organisms, right? So while we were discussing the catalase organisms, I have already told you something called as CGD. Can anyone tell, if you remember, what is CGD, guys? What is CGD, which I told you in the last class? Yeah. I've explained you very clearly regarding what is CGD and what is happening with the catalase positive organisms, right? So what is CGD? Excellent, Roy. Perfect. CGD stands for chronic granulomatous disease. Chronic granulomatous disease. CGD stands for what? CGD stands for chronic granulomatous disease. Don't worry, Hussain Raza, the video is already uploaded there, right? So you can watch them. Nothing to worry. So CGD stands for chronic granulomatous disease. Okay. So we discussed that previously, and we are about to discuss another important thing, that is pigment producing bacteria. Very, very important, guys. Now, after discussing this, you will see how many MCQs might have been framed from this. Pigment producing bacteria. Right? We shall be discussing pigment producing bacteria. Right. So in this pigment producing bacteria, the first bacteria which we shall be discussing is Actinomyces israeli. Right. I hope all of you know what is Actinomyces israeli, right? I hope all of you know what is Actinomyces israeli. So Actinomyces israeli, right? Basically, we basically say that the sand in Israel is yellow in color. So yellow, like yellow granules, you know. So that is the reason why Actinomyces israeli is yellow color, and it releases some kind of granules. These granules are called as sulfur granules. So if you look at the specimen of I'll show it to you in a minute. If you look at the specimen of that, the specimen looks yellow in color. Okay, so it is gonna release what? Yellow sulfur granules. Yellow sulfur granules. You know, Actinomyces israeli is so dangerous that it will cause abscesses on the face. It will cause abscesses on the abdomen. Okay, if I take that abscess, listen very carefully. If I take that abscess, put it under a microscope and add some stains and all, and look at that, you know how does it look? It looks like this. This is how Actinomyces israeli is gonna look. You see? Yeah, this is how Actinomyces israeli is gonna look. So these are the yellow sulfur granules which you can see. Okay, so this is nothing but your Actinomyces israeli. So all these are your Actinomyces israeli over here. Okay, see guys, today I'll be teaching you the most important topics, and I'll also make you to do the MCQs as well. So what I want from your side is you just have to be with me. Okay, don't write any notes. So right, okay, if you want you can write notes, but when I tell you to stop writing notes, please stop writing notes. But important thing is that we are going to discuss very important topics, guys, very important topics. Okay, right. So look here now. So one thing we are done with that. One thing we are done with that. What is that? Actinomyces israeli, yellow sulfur granules. Right now, the next one, Staph aureus. That is Staphylococcal aureus. Staphylococcus aureus. You know what is Staphylococcus aureus? Staphylococcus aureus, aura. You know what do you mean by aura? Aura means gold. Aura means gold. So that is the reason why this Staph aureus is producing some gold color colonies. That is why they have named it as Staphylococcal aureus. So they are producing what? Gold color colonies. Gold colonies. Gold colonies. If you want, I'll show you. You see here. Yeah. I hope this picture is clear for you. You see the gold color colonies there. These gold color colonies are produced by Staph aureus. Okay, these gold color colonies are produced by Staph aureus. See, as I already told you, microbiology is a dry subject. So these are the factual points which you have to remember. But as we are proceeding in the next few pages, you will listen to a number of wonderful concepts, guys. Believe me. Third important thing is that we have got something called Pseudomonas aeruginosa. Now tell me, which pigment does this Pseudomonas aeruginosa produce? Pseudomonas aeruginosa. Very good. Roy, excellent. Pseudomonas aeruginosa produce green color pigment. Green color pigment. Very good. Medical love, Dr. Bentek. Excellent. Pseudomonas aeruginosa will produce green color pigment. And let me tell you one thing, guys. Let me tell you one thing. By chance, if a patient has got Pseudomonas aeruginosa infection, I'm telling you, the entire wound, when you look, it is completely green in color. You know, that is very interesting, guys. See, this is how Pseudomonas aeruginosa look. Pseudomonas aeruginosa looks green color. Okay. And second important thing I would also like to add here is that whenever you see a patient of this Pseudomonas aeruginosa, the wound looks completely green. So I don't have a picture right now, but I'll put that picture. I'll put that picture as a question. Okay. So Pseudomonas aeruginosa infections, they are very suppurative, guys. They produce green color pigment. Even on the wound also you'll see green color pigment. Okay. Now, the question which has been asked previously is that what makes this Pseudomonas aeruginosa look green? Why is it releasing green color pigment? Because because it basically releases two important things. What are the two important things it releases? It releases pyocyanin. It releases pyocyanin. Not only that, it also releases pyoverdin. Pyocyanin and pyoverdin. Both this pyocyanin and pyoverdin, they impart green color to the bacteria or the pus, whatever it is discharging. Right? Roy, you're perfect. We call it pyocyanin pigment. Pyo is pus, cyan means green. Excellent, Roy. You're perfect. Excellent. Very good, Dr. Bentek. It is verdin and pyocyanin. You're right. You're perfectly right. And the last one to remember is the last one is Serratia marcescens. I'm sorry if I spelled it wrong, but Serratia marcescens. So how do you remember it? How do you remember it is that Serratia has got two R's, R for red. Okay, I know this is not better mnemonic, but you have to try something. Right. So this is a red color pigment. Now this is how Serratia looks. Look at this. Look at this pigment here. This is how the colonies of Serratia look. Okay. This is how the colonies of Serratia marcescens look. So Roy told me here Mars, in the sense it is a red planet, so they named it as red. Very good, Mars. Excellent. So guys, is it difficult for you to remember these four things? Right? Actinomyces israeli, yellow sulfur granules, Staph aureus, aura means gold, that is why gold colonies. Right? Pseudomonas aeruginosa, that everyone know that it is green color pigment. Serratia marcescens, that is red color pigment. Okay, so you told me, are there any MCQs? Yes, there are MCQs. Either they can ask you direct MCQs of what color it is releasing. Second thing is that they can put the entire thing in a single multiple choice question. How is that clinical multiple choice question asked? All of you just concentrate here. Look here now. Now you will answer me. Okay. Who will answer me is, let us say, someone was having problem with microbiology. Rupshika Datta, are you here? Yeah. So Rupshika Datta will answer me this, that a 37-year-old woman, right? A 37-year-old woman presents to her primary care physician after returning from her honeymoon. Okay, in Cancun. A few days ago, she began to notice an eruption of small red color bumps that cover her torso, back, and buttocks. Okay, there are some red color bumps on the back side, on the buttocks, etc. Okay, the patient first thought she had acne. No, that is usual, but became worried when the rash continued to itch. Culture yields. You see, what happened is that when she keeps on itching, there will be a little bit discharge coming out. So I will take that with a swab and I'll put it under the microscope and I'll look at, then I find a green color pigment. Now, what is the organism? Now, from the options given below, what can be the organism, guys? Saumya Deep, very good. Excellent, all of you, very good, guys. Priyanshi, excellent. Sadia Ansari, perfect. It is Pseudomonas aeruginosa. Pseudomonas aeruginosa, right? Let us do another question. And after that, I'll tell you from where these questions were asked. Okay? Harshil Choksi, I'm sorry, you're right. Okay, everyone now concentrate on the questions there. 52-year-old man is brought to the emergency department after being found down on his sidewalk. On presentation, he is found to have overdosed with opioids. So he is given naloxone and quickly recovers. Okay. Physical exam also reveals there were what? There were lumps. Where are the lumps? Lumps on the neck, lumps on the face. Okay, uh, that are covered by small yellow granules. So we are not sure what are those yellow granules. Okay, as shown in the image below. So in the image they have shown some yellow granules. So this is how it is. Look here now. What were they asking is, these lumps are slowly draining yellow pus like fluid. Okay, see, yellow pus in the sense golden yellow pus, that can also be staph aureus. We are not sure about that. He says that these lumps have been present for several months but has been ignored because there were no fever or pain. Okay, he does not recall the last time he visited a primary care physician. So on the oral cavity there were multiple abscesses and cavities. So he is asking the most likely cause. Medical love, it is not Pseudomonas. Pseudomonas is green, right? Small yellow granules is the point here, and the answer would be what? Answer would be Actinomyces israeli. I also told you Actinomyces israeli causes abscesses on the face, on the abdomen, right? So if you can see here, there are multiple cavities and abscesses. Okay, remember that. See guys, two things to remember: yellow sulfur granules, multiple abscesses. Directly put an answer as Actinomyces israeli. Okay, directly put an answer as. Very good, Suchit. Suchit, excellent. Right. So shall we continue with the topic? So far, guys, so far, did you understand? Is there anything difficult? Right. I know it would be difficult to remember, but you have to try it. Yeah. All clear? Shall we proceed? Yeah. Okay, fine. So look here, guys. Thank you. Right. So look here. In bacteriology, whichever bacteria you study, okay, whichever bacteria you study, there will be some things like it is a gram positive organism, it is a catalase positive organism, it is an optochin sensitive organism, it is a beta hemolytic bacteria, or sometimes it is a alpha hemolytic bacteria, it can also be it is a gamma hemolytic bacteria. So these are a couple of things which you definitely have to know. For every bacteria, these are like identity cards. Okay, you definitely have to know them. So I'm going to teach you hemolysis. Hemolysis. Now, medical love, yes, I got your answer. I already read your answer. You were right. Hemolysis. Hemolysis is basically of three types, guys. Now, one is called as alpha hemolysis. Another one is called as beta hemolysis. I hope all of you have heard this. You might be confused what is this. And another one is called as gamma hemolysis. Alpha, beta, and gamma hemolysis. Now look here. What is alpha? What is beta? What is gamma? I'm going to teach you. What you do is that you take an agar plate. Okay. I hope all of you have seen this agar plate, right? So it is somewhat like this. You take an agar plate. Now within this agar plate, what you do is that you add a lot of sheep blood. I mean, you don't add a lot. Actually, you add how much it is sufficient. So this is the sheep RBC which you have added. Blood in the sense RBC, right? So what you have added? You have added sheep RBC. You have added sheep RBC onto the agar plate. Okay. Now what I do is that I put a bacteria called beta hemolytic bacteria, whatever it might be. What is the example? I'll tell you later on. But I'll put a bacteria and the bacteria is named as beta hemolytic bacteria. Let us say that you see this blue color bacteria which I have put in the center. This is a beta hemolytic bacteria. You know what will happen? This beta hemolytic bacteria, it starts releasing some toxins outside its body. You know what are these toxins? This is called as beta hemolysin. Beta hemolysin. And the capability of beta hemolysin is so powerful. This toxin is so powerful that whatever RBC are located surrounding it, it will break down all the RBC. Okay. Let us say surrounding this bacteria, there are 100 RBC. surrounding this bacteria, there are 100 RBC. Now this beta hemolysin has a capability to break down all the 100 RBC. All the 100 RBC are broken down. So when all the 100 RBC are broken down, there will be a zone there, a death zone surrounding the bacteria, isn't it? So can I tell that this is how the death zone looks? You see, surrounding the bacteria, there is no RBC. Complete hemolysis has happened. Complete breakdown of RBC has happened. This is called as beta hemolysis. So beta hemolysis is also given another name called as complete hemolysis. Complete hemolysis, right? So practically to tell you guys, I don't want to confuse you. Right, look at this picture. Look at this picture, guys. I know it is a little bit blur, but look at this picture. The center one is the bacteria. Center one is the bacteria. Okay. Now, surrounding the bacteria, there is a zone of hemolysis. I mean, no RBC surrounding the bacteria. There is a white, bright zone there, isn't it? Now, this zone, you call it as zone of hemolysis. Zone of hemolysis. So from today onwards, if I show you a slide of a beta hemolytic bacteria, will you be able to identify, guys? Yeah, I don't use gray. I don't use gray at all. I use black. Right. So will you identify, guys? Will you identify, yes or no? Very good. Very good. Excellent. Excellent, guys. Let me tell you one very interesting thing here. Let me tell you one very interesting thing, that one of the example of beta hemolytic bacteria, right, is Staph aureus. Now, how are you telling it? Can you prove? Yes, I can prove. I told you in the starting, Staph aureus will produce golden color colonies. Look at these colonies now. How are they? Aren't they golden color? Yeah. Aren't these colonies golden color? They are golden color colonies. So this bacteria, this slide which I've put in front of you is a slide of Staph aureus, right? Clear now? Right. Very good, guys. Excellent. Excellent. Nice. Now let us switch on to the next bacteria, that is alpha hemolytic bacteria. Hemolytic bacteria. Alpha hemolytic bacteria. Okay. Simple thing. I'm not drawing the entire petri dish and all. So what I'll be doing is that I'll be taking a bacteria. So let us say this is alpha hemolytic bacteria. Okay. What is this bacteria? This is alpha hemolytic bacteria. Now, surrounding this alpha hemolytic bacteria, I have put 100 RBC. Sannyasi, I use red, I use pink, blue, black, green, dark red, and dark black. Okay. Right. So in the screen, it might look gray for you, but I don't use gray actually. Right. So outside this alpha hemolytic bacteria, I have kept 100 RBC. Same thing. Alpha hemolytic bacteria also will release a toxin called as alpha hemolysin. It will release a toxin called as alpha hemolysin. But you know, compare beta and alpha hemolysin is little weak. So when I tell it is little weak toxin, instead of destroying all the 100 RBC, it will destroy only 50 RBC. So only 50 RBC will be destroyed. See, only 50 RBC will be destroyed, and there will be some RBC left surrounding the bacteria. So can I call this type of hemolysis as partial hemolysis, or I can call it as partial hemolysis? Partial hemolysis. And you know one very important thing, alpha hemolysin will break down the RBC. Okay. So when RBC is broken down, RBC will release what? What will RBC release? Hemoglobin. RBC will release hemoglobin. Hemoglobin will be later on converted to, yes, biliverdin. Hemoglobin will be later on converted to biliverdin. Biliverdin is what? Which color is biliverdin, guys? Biliverdin is green color. Green color. Okay, that is the reason why alpha hemolytic bacteria, the colonies look green. Okay, so that is the reason why this is also called as green hemolysis. Green hemolysis. Let me show it to you. Let me show it to you. Right? Okay. Fine. You see this? Surrounding beta, there is a vibrant zone of golden color. We discussed, I'm not discussing it again. We are done. Come to alpha. Surrounding alpha also, there is some zone, a very minute zone, if you can see, right? Small zone here. I'm zooming the picture. You can see a small zone like this. Small zone here. Right? So this is alpha hemolysis, or alpha hemolysis is also called as incomplete hemolysis. Okay, and it is green. That is why green hemolysis. The last one which we will be discussing is gamma hemolysis. Same to same, even in gamma hemolysis also, a toxin is released that is called as gamma hemolysin. Gamma hemolysin. Beta hemolysin is more powerful than alpha. Alpha is more powerful than gamma. Remember it as bag. BAG, bag. Beta, alpha, gamma. So it means guys, by now I'm very sure that you might have known that surrounding this alpha, surrounding this gamma, if I put 100 RBC, do you think all the 100 RBC will be destroyed? Yeah? No. Not even a single RBC will be destroyed. Not even a single RBC will be destroyed because gamma hemolysin is too weak. So it means it is not at all causing any kind of hemolysis. So that is the reason why I call it as incomplete hemolysis. Incomplete hemolysis, right? Incomplete hemolysis. So look at this. This is a gamma bacteria. Surrounding that gamma, do you find any zone of inhibition, guys? No. Surrounding that gamma, very closely attached to gamma bacteria, there are very good bacteria. I mean, very good RBC there. There is no zone of hemolysis, right? So these are the important things which you need to keep in mind. Okay. Now let us discuss the examples. Okay. Examples like, for example, alpha, beta, gamma. Okay. Alpha hemolytic bacteria. Streptococcus pneumoniae is an alpha hemolytic bacteria. Not only that, alpha hemolytic bacteria are also another group called as Viridans streptococci. Viridans group of Roy, excellent Roy. Viridans group of streptococci. Okay, Viridans group of streptococci and Streptococcus pneumonia. Right. Within this Viridans group, we have got two things. What are those two things, guys? Anyone? What are the two bacteria which are located within this? Simple way I'll tell you. Look here. V stands for violence. Okay. V stands for violence or violation of rules. So let us say this house is completely violated with rules. Okay. What are the rules? That you should not play songs. Okay. So whenever you're violating rules, what will happen is that you might mute songs. Whenever I violate you, you will mute the songs. You might mute songs. But when I go out, you will play the songs. But temporarily, you might mute the songs. Now, what I'm trying to tell you, MI stands for mitis. Streptococcus mitis. MU stands for mutants. What is this mutants? Streptococcus mutans. And SO stands for sanguinis. Streptococcus sanguinis. These are the three things that are, yeah, these are the three things that are grouped under Viridans streptococci. And one wonderful point I'm going to tell you, that all these three bacteria are located in your mouth right now. In you and me, in everyone, we have got this bacteria. Okay, right. Now coming to beta. Coming to beta. Right. Now, do you know what is meant by BAP? BAP means BAP. BAP means father, right? In Hindi we call it as father. Now, what does A stand for? A galactiae. A galactiae. And what does P stands for? Pyogenes. Streptococcus pyogenes. Clear? Yes. Hasnain Raza, these are normal flora. These are normal flora. I'll be telling you these about these flora. I'll give a very wonderful explanation of this flora. How the first bacteria, when we enter into the bacteria and all, I'll be teaching you each and every bacteria in detail. That time I'll be telling you how this bacteria enters into your body, from there to the circulation, and from there how it damages your body. Everything I'll put up a big picture in front of you and I'll tell you, okay? But yes, these are the normal flora. Gamma. Gamma is last, right? Gamma is last. So, Enterococcus. Enterococcus is gamma. So, there are two types of Enterococcus bacteria. One is called as Enterococcus faecium. Another one is called as Enterococcus fecalis. Fecalis. Faecium and fecalis, both of them are different. Okay? Both of them are different. Right. So, till here, did everyone understand? Did everyone understood whatever I've taught you? Yeah. Be fast, guys, so that we need to fast up the things and we can clear microbiology entirely as soon as possible. I mean, not in this session, but in the subsequent lectures. Thank you, Gideon Riame. Gideon Riame, thank you. Priyadarshini, thank you. Right. Now let us switch on to the next important thing. Which color isn't visible? Which color isn't visible? Okay, I'm sorry. Green color isn't visible. Let me test it. Yeah, green isn't visible. So guys, I'm not using green. I'm really sorry. I'm really sorry. I'm really sorry. Now let us switch on to bacterial virulence factors. Yeah, I understand. Yeah, it seems white color. Bacterial virulence factors. So whenever you look at the bacterial virulence factors, the first important thing I would want you to look is that IgA is a bacterial virulence factor. Okay, is IgA bacterial virulence factor? Yeah. Anyone tell me, is IgA a bacterial virulence factor? Simply, don't confuse what is virulence factor, guys. See, for example, if I'm going for a war, I'll take all the, what you say, all the swords, all the arrows, bows, and all. Right. So all these are the virulence factors for me. So they're protecting me. So for bacteria also, there are some things that protect the bacteria. So can anyone tell? Can anyone tell? Is IgA a virulence factor? Yes or no? Very good. Dr. Benedict, very good. IgA is not a virulence factor. IgA is a first line defense, right? Which is present within your mouth, within your saliva, isn't it? So IgA is found in secretions. It is found in secretions, mainly the mucous surfaces. Mucous surfaces. And this is called as the first line of defense. First line of defense. Then why are you teaching us bacterial virulence factors? See, bacteria has got IgA protease. Bacteria has got what? An enzyme called IgA protease, which will enter into your mouth. You take the bacteria inside, that bacteria will release IgA protease, and this IgA protease will break down that IgA. So it will break down the IgA in your mouth. So when IgA is broken down, do you think the first line defense is still there? No, the first line defense is gone. So in this way, bacteria is surviving. It means IgA protease is a bacterial virulence factor, isn't it? All right. Now, which one seems white? Oh, again. Give me a second. Give me a second. I'm really sorry. Give me a second. Right. I hope this is clear. Right. So this will be your first line of defense. First line of defense. Right. Now look here. All of you look here. So IgA protease is a what? IgA protease is a virulent factor, right? Now, which group of bacteria is having IgA protease? Remember by the mnemonic shin. Now, what is shin? All of you know what is shin, right? So, S stands for Streptococcus pneumoniae, right? H stands for H and I. H and I, both of them stand for shin. Streptococcus pneumoniae, Streptococcus pneumoniae. And what does H and I stand for? H and I stand for Haemophilus influenza. Haemophilus influenza. Haemophilus influenza. And N stands for Neisseria. So guys, there are three bacteria which releases IgA protease, that is Streptococcus pneumoniae, Haemophilus influenza, as well as Neisseria gonorrhea. So you understood this. This is the first. What is the first? The first important thing is that IgA protease is a factor, is a bacterial virulence factor. And the second bacterial, let me make a box here, guys. Let me make a box. Second important virulence factor is that, look here very carefully. Look here very carefully. The second one, this is a bacteria. Okay. On the surface of bacteria, there are some protein molecules here. On the surface of bacteria, there are some protein molecules. Okay. Now, what these protein molecules will do is that whenever a phagocyte will come, so this is a phagocyte like this. Okay. Now, whenever this phagocyte tries to engulf that bacteria, these protein molecules will kick the phagocyte. Then the phagocyte will go away. Okay. So, it is responsible for preventing phagocytosis. And what is this virulence factor name? It is called as protein A. Protein A. So protein A helps to prevent phagocytosis. Phagocytosis. Clear? It helps to prevent phagocytosis. So protein A. And third important factor is called as M protein. Now there is a very important concept I want all of you to pay attention on this, guys. What is M protein in the sense? Look here. This is a bacteria like this. This is a bacteria like this. On the surface of the bacteria, I have got some protein molecules like this, and these protein molecules are called as M proteins. M proteins. Okay. So let us also tell that this is the heart. This is the heart. Let us also tell that this is a joint like this. This is a joint like this. So even on the surface of the heart, even on the surface of the joint, even on the surface of the heart, even on the surface of the joint, we have got M proteins. All of you pay attention now. The M proteins which are located on the bacteria are structurally and functionally similar to the M proteins which are located in your heart and on your joints. Okay. Now, this bacteria has entered into your body. When this bacteria will enter into your body, your antibodies will go and attack that bacteria. So, antibodies don't know anything. Antibodies know only one thing. There is something called M protein which came into our body. We should go and attack. Now, hundreds and hundreds of antibodies will come and they start attacking wherever there is M protein. So, M protein is present on the bacteria. Bacteria is dead. But M protein is also present on the heart. So, if it attacks the heart, patient will have what? Endocarditis. Patient will have what? Endocarditis. Sir, endocarditis means inflammation of endocardium. So, will the antibodies penetrate inside the endocardium and cause? No. First, there will be pericarditis. Outer layer is gone. First, there will be pericarditis. Then there will be myocarditis, and after that, there will be endocarditis in severe cases. Then what about the joints, sir? The antibodies will also attack your joints. So when the antibodies will start attacking your joints, this will lead to arthritis. This will lead to arthritis. And this kind of mechanism, which the bacteria is, you see how cunning the bacteria is? Bacteria is entering into your body and making your own body cells to kill your body. Isn't it? So this type of cunningness, whatever you see, the cleverness of this bacteria, right? This is called as molecular mimicry. Molecular mimicry. Did everyone understood what is molecular mimicry? Obviously, it would lead to rheumatic fever. Obviously, it would lead to rheumatic fever. Very good. Yes, Osama Sheikh, it is acute rheumatic fever. Very good. Very good. But I don't want to discuss that, because if I discuss again, I am going into medicine. I don't want to go that deep. Okay. But you are right, guys. Perfect. Priyanshi, you're right. Excellent. So this is called as molecular mimicry. Did everyone understood the virulence factors? The third virulence factor which we discussed was M protein. M protein. So these are the three virulence factors which will be asked in your exams, right? Very good. Now we shall be discussing one of the most important, most difficult, most confusing topic in microbiology, that is your staining methods. I don't understand why students find difficult. In fact, when I was studying my MBBS, even I was having a lot of difficulty in this. In fact, I never studied microbiology, but you know, I'm good enough to teach you. But so staining techniques, is everyone ready with the staining techniques? After we discuss the staining techniques, we shall have a small recess. Okay, coming to the staining techniques, look here, everyone, pen down, pen down. If you want the concepts to enter into your mind, just pen down. Right, coming to staining techniques, guys, simple thing. Why am I staining a bacteria? Because I can't see bacteria from my naked eyes. Okay, then when you stain, can you see with your naked eyes? No, I should put it under a microscope. Okay, bacteria basically are colorless, guys. When I add some color to the bacteria, then the bacteria will glow. For that, I need staining techniques. First important thing is that to impart color to the bacteria. Sumit Paswan, you are perfectly right. You're perfectly right. M protein is also present on mitral wall, and moreover, endocarditis, right? Endocarditis, whenever we discuss endocarditis, the mitral valve is more affected than the tricuspid valve. Okay, so he's right. But we are not going that deep, Sumit. You're right. Shine Khan, thank you so much. So to impart color, guys, all of you, look here. So what will I do? I will take an agar plate. I will add some bacteria. Now after adding some bacteria, I will also add some color. Okay, I'll put it under a microscope. Now when I look from the microscope, I can see the bacteria very clearly. But important thing is that, why am I doing this? I am doing this to study the structure of the bacteria, to study the flagella of the bacteria, to study the pili, the fimbriae, right? To study the cell wall. So to study all the structures of the bacteria, I'm staining it. But if the bacteria is keeping on moving in the slide, will I be able to fix my eye towards a single bacteria? No. So what I need to do, instead of fixing my eye, I will fix the bacteria to the agar plate. So bacteria is giggling, it is moving, moving, moving. What I'll do is that I will take that plate, I'll put a lighter below and I will heat it a little bit. When I heat it a little bit, the bacteria will fix to the surface. This is called as fixation. So the next important thing is that you need to do fixation of the bacteria. Not only adding color, fixation is important. There are two types of fixations. There are two types of fixations. Either by heating, this is called heat fixation, or I will add a chemical called methanol. This is called methanol fixation. Heat fixation, methanol fixation. Okay. In heat fixation, in the sense, don't heat it for 1000 degrees or 200, 300 degrees. You have to heat it very mildly. Okay, gentle flame you have to keep. Okay, sir. Fine, very good. Then methanol fixation is done for what? For blood smears. Any kind of blood smear you take, in that blood you have bacteria. Bacteria is moving, swimming in the blood. So you add methanol, bacteria will stick to the surface. Okay, right. So overall, I wanted to discuss a classification called as type of stains. From here the topic starts. There are three different types of stains, guys. There are three different types of stains. Okay, what are these? These are, one is called as simple stain. One is called as simple stain. Another one is called as differential stain. Differential stain. Yeah, guys, someone has commented that in drug users, in IV drug users, tricuspid wall effect will be more. You're right. Why did you delete your comment? You are perfectly right. You, in IV drug users, where do they inject, guys? Where do they inject the injection, whatever it is? To the veins, right? To the veins they inject. Let us say a bacteria has entered into the vein. All the veins will connect and drain into which heart? Right or left heart? They drain into inferior vena cava. From inferior vena cava, they drain into right heart. Right heart is having mitral valve or tricuspid valve. Yeah, tricuspid valve. So firstly, the bacteria will enter and hit the tricuspid valve and cause endocarditis there. Okay, very good, fine. We are not, I'll discuss medicine very detailedly later on, guys, but right now we are concentrating on the micro, so please don't distract me. So simple, differential and special stain. Simple, differential and special stain. We shall discuss in very clearly after this class. I'm telling you that there won't be any need for you to revise. What is simple stain? What is simple stain? Simple stain means I will add only a single color. That is simple stain. Simple thing. Okay. I will add only one color. Only one stain. That is called simple stain. So whatever bacteria is there, that will take up that color. It will glow. You can see. So can you tell me an example? I did not understand it clearly. Simple thing. I will either use violet color or I will either use pink color, but I will not use both of them. I'll use any one of them. Okay. I will either use methylene violet, which will impart violet color. I will either use saffronin, which will impart pink color, Okay, crystal violet and pink. Very good, Priyanshi. Madhavan Kuti, thank you. Yeah, you are the one, right? Perfect, Madhavan. Your answer was right. So this is simple stain. Okay, look here. Can anyone tell me, did I use pink or methylene violet here? Did I use pink or methylene violet? I have used what? I have used methylene violet. I have used methylene violet. Right? So this is methylene violet. Methylene violet. So this is called simple stain, guys. So Katam, simple stain is done. Next, differential stain. What is differential stain? What is differential stain? Differential stain means if I use different colors, it is called differential stain. If I use a single color, it is called simple stain. If I use more than one color, it is called differential stain. So more than one stain is called differential stain. Okay. So simple thing is that I brought chocolates, I brought biscuits, I bought cakes, I bought pancakes, I bought let us say fruits, right? So all of them I've gave it to you. So some group of bacteria love chocolates, they take up that chocolate. Some of them love biscuits, they take up that biscuits. So when I use differential stains, bacteria, whatever color they want, they will take up. For example, gram-positive bacteria love violet, they will take up violet. Gram-negative bacteria love pink, they will take up pink. Right? So if I use more than one stain, different bacteria take different colors. I mean, different bacteria take different stains. Stain means color, guys. Okay. So why are you telling me this? Why am I telling you is that only by that way I can know which bacteria is that. If I see a violet color bacteria, then I will fix in my mind that it is gram-positive bacteria and I will start giving medicines against that. Okay. So this is useful to differentiate different organisms. Excellent, Priyadarshini. Priyadarshini has given me an example. You're right. Yes, there are two examples of differential stain. One is called as gram stain. Another one is called as Zn stain or Zeal-Neeson staining. Zn stain. Okay. Now we shall discuss gram stain and Zn stain. Now, all of you guys, pen down. Don't write anything. Let me discuss gram staining now. Gram stain. Simple thing. All of you, look here. Simple thing. I will take a agar plate. Okay. After taking agar plate, I will add some bacteria. Now, after adding some bacteria are there. What will I do? First thing I will do is what? Fixation. First thing I will heat it and fix the bacteria to the surface. So the first step in gram staining is fixation. Okay, fixation is done. Now what I will do is that I will pour some color. Okay, I will pour a color called as gram color, or shortly I can call it as GR color. Shortly I can call it as GM color, gram staining, right? GM color or GM staining. GM color or GM staining. Now what is this GM color or GM staining? G stands for gentian violet. G stands for gentian violet. And M stands for methyl violet. But whatever it is, I'm adding violet color, right? Okay, I have added violet color. What will happen, guys? Anyone tell me? Can anyone tell me what will happen? You know what will happen? Within that, I have added two types of bacteria. I told you. Gram positive I have added, gram negative I added. Gram positive will love violet. Gram negative will hate violet. So whenever I add violet color, whenever I add gentian violet, gram positive loves violet. So it will drink up all the violet color. Gram negative does not like violet, but what it will do is that it will take that violet color and rub over its body. One is drinking, one is rubbing over its body. So both of them are colored now. So what I'll do extra is that, I will add a chemical so that the color will fix to the bacteria. I don't know whether the color has fixed to the bacteria. So I'll add a chemical so that the color will directly enter into the body of the bacteria, and that chemical you name it as mordant. What is a mordant here? The mordant is iodine solution. What will it do? It will fix the dye to the bacteria. Clear? Okay, fine. So after adding mordant, what will you do? What will I do is that now I will add something called as decolorizer. When I add decolorizer, when I add Priyanshi, you're right. When I add decolorizer, you know what will happen? What will happen is that gram-positive bacteria love violet, right? So even after adding decolorizer, gram-positive bacteria will not give away that violet color. It will tell, I won't give you the violet color, I want it. But gram-negative bacteria is so happy because it doesn't like violet. The moment you add decolorizer, gram-negative bacteria will swipe up all the violet color which is attached to its body. Okay, so when you add decolorizer, for example, decolorizer like acetone, acetone is a decolorizer. Let us say ethyl alcohol is a decolorizer. Let us say acetone alcohol is a decolorizer. Whenever I add decolorizer, if there is gram-positive bacteria will not give away that color. So gram-positive bacteria will remain violet only. But if there is a gram-negative bacteria, it loves to give away that violet color. So it would become colorless. It would become colorless. So in this way, I can differentiate two important things, that is gram positive or violet in color, gram negative or colorless. Now a doctor comes and tells, now if gram negative is colorless, it doesn't look nice, but can you please add some color to that? Yes. So what I'll do is that I will add pink color, some pink color reagent. Let us say safranin. I told you in the starting, safranin is a pink color reagent, right? Right? Along with safranin, I'll also add carbol fusion. Carbol fusion. Both of these are pink color agents. Now, the interesting thing you need to know is that gram-negative bacteria loves pink color. The moment you add pink, gram-negative bacteria will drink away that pink color and the colonies appear pink. Okay, so then you can differentiate it as gram-negative bacteria. Did you understand here, guys? Did you understand this explanation? If you understood this, Gel Nation will be very easy. Yeah? Right. Very good. So, you know what are the questions they will ask you in the exam? Simple thing. They will ask you in gram staining, what is the primary stain you used? So this is called as the primary stain. Primary stain. This is a question in your exam. Primary stain is either gentian violet or methylene violet. Second question is, what is the mordant do you use in gram staining? That is iodine solution. Third thing is, what is the decolorizer which you use? So either of these three decolorizers which you use. Next important thing is that what is the counter stain which you have used? Counter stain which I have used is pink. So this will be counter stain which I have used is pink. This will also be a question. Okay. All of you understood? Yeah. So these are the MCQs which will be asked from this, guys. Now, apart from this, very good. Apart from this, another important thing is that we are mainly focusing on gram-positive bacteria now. In the starting, in the first microbiology class, I have already told you that whatever has a thick peptidoglycan layer or a thick cell wall is called gram-positive. Whatever is having a thin peptidoglycan layer or thin cell wall is called gram-negative. Right? So it is having what? Thick PPG, peptidoglycan layer. PPG means peptidoglycan layer. Right. Okay, fine. PPG is peptidoglycan layer, right? I'll give you a question, guys. The question is that, for example, if the bacteria is too thin, if the cell wall of the bacteria, is too thin, too lean, do you think can it retain The gram positive color, yes or no? Very good. It will leak the dye out. No, Biki, you are right. It will not. So if the cell wall is too thin, if the cell wall is too thin or too lean, it will not retain the color. It will not retain the color, right? So T stands for treponema. Treponema pallidum, the cell wall is too thin. That is why treponema pallidum is not, it cannot retain the color, but it is a gram positive. Second important thing. Second important thing is that, for example, guys, another question. Cell wall is having peptidoglycan. Okay. This is a bacterial cell wall, and bacterial cell wall is having peptidoglycan. There is another bacterial cell wall. Instead of peptidoglycan, there is a lot of fat. There is a lot of fat. Okay. Now tell me, will the color enter into peptidoglycan or will the color enter into fat? Yeah. This is peptidoglycan. This is lot of fat. Okay. High lipid. This is highly lipid. Gram color will enter into peptidoglycan rather than entering into thick layer of fat, thick layer of lipid. Okay. So the cell wall should have thick peptidoglycan layer. That is rule of thumb. Other than that, whatever it is there, it won't. Right? So thick peptidoglycan layer will have the color, but this thick layer of fat, thick layer of high lipid will not retain the color. And let me tell you one thing. Let me tell you one very important point here. High concentration of lipid in the cell wall, right? You know, lipid in the sense, you know what? Mycolic acid. We have already studied long ago, many times, right? That cell wall is containing high concentration of mycolic acid. Now you might get a doubt, sir, you are telling mycolic acid, then why it is highly lipid? Mycolic acid is a free fatty acid. It is a fatty acid. Okay. So if there is mycolic acid, another stain I will use, that is called as acid fast stain. Acid fast stain. Or Zn stain. Zn stain. But gram staining will not work for this. Gram staining will not work for this. Okay. What is the example? All of you know the example? Yes. All of you know the example? Anyone? Anyone? Anyone give me an example, guys? Yeah, example of Zn stain. Excellent, Priyanshi. Mycobacterium. MTB. Mycobacterium tuberculosis. Excellent. Priyanshi, very good. Mycobacterium. Very good. Third important thing. Third important thing is that, for example, if the bacteria is having no cell wall, do you think if the bacteria is having no cell wall, do you think will it stain? Gram staining? Will gram staining work here? Yes or no? No. Very good. Biki, Priyanshi, you're right. No, it won't work. Right? Very good. Excellent. Nai. Soumyadev, Nai. Huh. Very good. So those bacteria which don't have a cell wall, they cannot stain. They cannot undergo gram staining. Okay, you know what are those bacteria? If I remove the cell wall, what is there? After the cell wall, what is the layer? Plasma membrane. So this bacteria ends with a name called plasma. For example, urea plasma. Okay. For example, mycoplasma, urea plasma, mycoplasma. So whatever is ending with plasma has no cell wall. So they also don't stain with the gram stainings. Okay. So I hope all of you understood this. I hope all of you understood this. Yeah. So we are done with all the MCQs that can be asked, guys. This is an MCQ. Right. This is an MCQ. Right. And this can be an MCQ. Right. Very good. Now let us enter into the Gill-Nielsen staining and then let us have a break. Okay, then let us have a break. Acid fast staining or Zn staining. Zn staining or acid fast staining. See guys, nothing has changed. Nothing has changed. Step 1 there, what did I use? I used a stain. That stain is called as primary stain. Here also I will use a primary stain. Primary stain. But before this fixation, okay, here also I will use primary stain. The primary stain there I used was what? Violet color. But here I will use pink color. What is that pink color? Carbol fusion. Carbol fusion, which will impart pink color. Okay, so what will happen? Let us say that these are acid fast bacilli, right? These are non-acid fast bacteria. So one is acid fast, another one is non-acid fast. Okay, when I add pink color, the acid fast will become pink, non-acid fast also will become pink. Both of them will become pink. Okay, but out of acid fast and non-acid fast, which bacteria loves pink color? Comment here, which bacteria loves pink color? Yeah, Priyanshi, excellent. Benedict, excellent. Acid fast bacteria loves pink color. Fine. After adding a stain, what did I do there? I have added a mordant. Here also I will add a mordant. There, what is the mordant I have added? I have added iodine solution. But here mordant, you know what will I add? Naveen Bhargav, good afternoon, good evening, I think. Good evening. Here, what is the mordant? Mordant is nothing but heat. I will heat it. When I heat it, all the pink color will fix very hardly to the bacteria. Mordant is nothing but heat. You know, don't heat it till the entire medium turns black. Heat it till the fumes appear. Okay, right. After that, there I have added decolorizer. Here also I will add decolorizer. But the decolorizer here I will add is H2SO4. When I add decolorizer, what will happen is that acid fast, non-acid fast. When I add decolorizer, what will happen is that acid fast bacteria love pink color, so it will not leave the pink color. But non-acid fast bacteria, they don't love pink color, so they leave away pink color. So in this way, you can differentiate between what is acid fast, what is non-acid fast. Sir, there you have added secondary stain also. I mean counter stain. Will you also add here? Yes, I will also add counter stain. But counter stain is not that much useful. You know what counter stain will I add here? Methylene blue. When I add counter stain, you know what will happen? Nothing is gonna happen, guys. Center you will see pink color bodies. Background will be completely blue like this. Background will be blue like this. Background will be completely blue, and on the background you will see pink color bodies. That's it. Okay. Look at the picture here. Now you'll understand very perfectly. Right. See here. In the center, I see pink color bodies, but background is what? Blue. What is this background I have added? Methylene blue, right? So, what is the background I have added here? This is your counter stain. This is your methylene blue. Background I have added methylene blue. And what are these? These are your acid fast bacilli. Okay. Now, sir, you gave me that one example. One example of acid fast bacilli is Mycobacterium tuberculosis. That's it. Are there any other things you want to tell me? Yes. Spores. spores. Second important thing, the head of the sperm. The head of the sperm is also acid fast because it has high concentration of mycolic acid. Third important thing, Mycobacterium tuberculosis. Fourth important thing, nocardia. Nocardia. Okay. No, very good, Priyanshi. Fifth important thing, parasites. You know what parasites? What parasites, Anyone? Anyone? What parasites? Yeah. Right. Don't worry. Parasites are crypto, crypto, cyclo, iso. Crypto, cyclo, iso. Crypto means cryptosporidia. Cyclo means cyclosporidia or simply called cyclosporia. Iso means isosporia. So, what are the three parasites here? Sporia, sporia, sporia. What are the sporia? Cryptosporia, cyclosporia, isosporia. Very easy to remember. Isn't it? Very, very easy to remember. Right. So, very good, all of you. So, what all we have discussed so far, guys? We have discussed so far, I'm just making up a map here. We have discussed so far that there is something called simple stain. We are done with that. We have something called differential stain. Differential stain. We discussed two examples of differential stain, right? One we discussed was gram staining. Another one we discussed was Zn staining. Zn staining. Now, when I tell Zn staining, don't think that Zeal Nason are the scientists who discovered this Zn staining. No, Zn staining was first discovered by Paul Ehrlich. Okay, later on they modified it. Later on they modified it. Okay, so simple staining we used methylene blue, methylene violet, right, and saffronin. So we are done with two things. Now we come across another important thing, that is special staining. We come across another important thing, that would be your special staining. Now let us take a break for 5 minutes, guys, and then we shall discuss this special staining. Okay, break. Right, so let us take a break. Right, guys, welcome back, welcome back, welcome back. Now, 20 members have gone for break. Hello, 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 come back, come back. Welcome, Benedict. Osama Sheikh, hi. Guys, are you all uh, see even whenever I'm explaining these things seem difficult to me. But are you understanding this, guys? Yeah? Are you understanding them? Suchit, hi. Joshu, namaskar. Thank you, Benedict. हाँ बिकी ये डिफिकल्ट टॉपिक है यस दिस इज लिटिल बिट डिफिकल्ट टॉपिक आई नेवर टच्ड ड्यूरिंग माय एमबीबीएस बट यू नो आई हैव टू टीच यू सो आई हैड टू स्टडी दिस yes Hasnain, I will summarize it. Definitely I will summarize it. Definitely I will summarize it, okay? Not like Dr. Najib, don't compare that legend with me, but I'll summarize like me only, okay? Right, so shall we start? This is not white bottle, this is a green bottle. Right, let us start now. Start now. Wherever you go in the world, you come across three staining methods. Okay? Joshua, yeah. When all my hair goes away, then you can call me Chota Dr. Najib. Right. So when right. So wherever you go, staining methods are three. Okay. So one is called as a simple staining method. Another one is called as a differential staining method. You use one color, you use more than two colors, and the last one is called special staining method. I'll discuss this so specially with you guys. Really, you'll like it. Special staining method. S for saint. You know, saints are special people because they don't marry, right? Right. So if you have an idea or if you are thinking to become a saint, then that is well and good. Saint. S stands for in saint. S stands for special staining. And what does A stands for? A stands for Albert staining. Albert staining. Okay. And what does I stand for? I stands for impregnation staining. Impregnation staining. And N stands for negative staining. Negative staining. First, let us start with impregnation. Then we'll discuss Albert because they are integrated with each other. Okay. So this is the overall classification of staining. Overall classification of staining. Now we shall be discussing impregnation staining. What is impregnation? Impregnation is not making someone pregnant. Impregnation staining is different, guys. Okay. Impregnation staining means, for example, I wanted to study the flagella of the bacteria. Okay? I wanted to study the flagella of the bacteria. But when I looked at that bacteria's flagella is too short. I can't even study. Now, if it is too short and I can't even study, what should I do? Should I tell the bacteria, please grow your flagella? No. So what will I do is that what will I do is that, let me write this thing for bacterial cells as well as flagella. Bacterial cells as well as flagella, which are too thin that I cannot study the structure within that. What would I do is that I will add some salt. Okay, I will add a salt called as silver salt. So whenever I add some kind of silver salt onto the bacteria, what will happen is that the salt will fall within the bacteria. The salt will fall within the bacterial cell wall. The salt will fall within the flagella of the bacteria, right? Very interesting concept. This is the flagella like this. So whenever I'm adding salt, what is happening is that within the bacteria, the salt gets embedded inside. Within the bacterial flagella as well as the cell wall, the salt gets embedded inside. Now, so what is salt, guys? It is a solute, right? So whenever I add salt, what will happen? Will the water from the bacteria go outside, or water outside the bacteria come inside? Tell me. Will it come inside or will it go outside? Will the water come inside or will the water go outside? No, Bicky. Osmosis, not the channel. Inside, very good. Soumya Dev, inside. Inside the concentration is too high. To dilute that concentration, all the water will enter into the flagella. So when all the water will enter into the flagella from the surrounding medium, don't you think the flagella will swell? The flagella will swell. So there will be swelling of flagella. So whenever there is swelling of this flagella, then I can study the flagella very clearly. I can study the centromeres, I can study the triplet arrangement, nine arrangement and all, everything. Okay. No worries, Biki. This is a little confusing. Even I was confused just before the lecture. So there will be swelling of the flagella. But whatever you are adding, what I'm adding? I'm adding silver salt. That is the reason why this method is called as silver impregnation method. Silver impregnation method. Silver impregnation method. The most common salt which I will add is silver salt. Most common salt which I will add is silver salt. Why are you telling this? This was asked in the exam, FMG exam. This was the question which was asked. Okay. So whenever I add when I use this silver impregnation method and when I add the silver salts, what will happen? All the structures, whatever are there, they become thick. So when they become thick, I can study. Okay, so this silver impregnation method is basically used to study two different types of bacteria, guys. Okay, so what are they? Haris Khan, hi, hello. So what are the two important bacteria here? One is called as H. pylori. Okay, and also another group that is called as spirochetes. Okay, if I use these silver salts for H. pylori, now that method is called as Warthin Starry stain. Warthin Starry stain. If I use the same method for spirochete, if I add this silver salts for spirochetes, that staining method is called as Fontana stain. Fontana stain. Fontana stain. Okay, sir, how do I remember? It is very difficult because I might write H. pylori to Fontana and spirochete to Warthin Starry stain. Simple thing, guys. Whenever you go for a war, right? How will you go? You go by walk or you go by helicopters. Obviously, you go by helicopters and land the bombs onto the soil, isn't it? So H stands for Helicobacter. Helicobacter means helicopter. So you go to war on helicopter, right? So that is why it is called as Warthin. I mean, it is that is not the reason why it is called Warthin, but this is how you remember it. Okay, this is how you remember it. Very important MCQ. Very important MCQ. Okay, very good. Next important thing. We are done with what? Impregnation staining. Now I'm discussing Albert staining. Albert staining. I'm sorry. Albert staining. What is Albert staining, guys? Why do I use Albert staining? Why do I use Albert staining is that I hope all of you know that in Clostridium diphtheria, in all the clostridial species, you have got some granules inside them. Let us say this is a Clostridium bacteria. Okay, Clostridium bacteria. Within this Clostridium bacteria, I have got some minute granules. Now these small granules, Roy, you are perfect. It is phosphate specific stain. Very good, Roy. These small granules are called as metachromatic granules. Metachromatic granules. Okay. So, metachromatic granules are also called as volutin granules. Volutin granules. And these granules are rich in phosphate. Rich in phosphate. Rich in phosphate. Whenever you add Albert stain, that stain chemical nature is in such a way that wherever there is phosphate, this stain will go and attach to that phosphate, and it will form a very strong bond that it won't even separate. So phosphate is located in metachromatic granules. I use Albert stain. Stain will go and attach to this metachromatic granules, and I can very clearly visualize them. Okay. So where do I have metachromatic granules? I see in Clostridium diphtheria. I see in Clostridium xerosis. I see Gardnerella Gardnerella vaginalis, which means vagina in the garden. No, this is a bacteria. Gardnerella vaginalis. And spirillum, that is a spirochete. That is a spirochete. So these are the organisms. These are the organisms where I would see metachromatic granules. Sir, can you please show me these metachromatic granules? Yes, I'll show you. You see this? You see these black color bodies over here, small black color bodies, which gives a tennis racket or a drumstick appearance, right? So these are called as metachromatic granules, and these are the granules of the Clostridium species. Granules of what? The Clostridium species. दिल गार्डन गार्डन हो गया भाई लोग. Right. Albert staining is done. Impregnation is done. Now let us go to the negative staining. Very important, guys. I don't know how important all these stains, but negative staining is super important. Why? I will tell you. Negative staining. Negative staining. Okay, fine. Negative staining is done mainly for capsule. I have discussed long ago regarding in this live session that surrounding the bacteria we have got capsule. Capsule is very strong antiphagocytic. I told you. Capsule is not stained by any stain. Okay, capsule cannot be stained. So look here very carefully. This is the plate which I have taken, agar plate. Okay. When I have added negative staining, you know what happened? What happened is that the bacteria will stain. The bacteria will stain. Surrounding the bacteria, what is there? I told you, surrounding the bacteria there is capsule, right? Here also the bacteria, and surrounding the bacteria there is capsule. Let us say here there is some empty bacteria. Surrounding the bacteria there is capsule. Here also some empty bacteria. Surrounding it there is capsule. Now I have added negative stain. When I added negative stain, what will happen is that the bacteria will stain and the background will stain, but the capsule will not stain. Okay, the bacteria will stain like this, and the background, see, this entire background also will stain black, but the capsule will not stain. So if I show you only, you'll understand, guys. Look at this. Look at this stain. The bacteria in the center stained, the background stained, but the capsule did not stain. Right? So this gives us a halo capsule surrounding the bacteria. So from today onwards, guys, if I ask you what is the best stain used for staining the capsule, that is your what? Negative staining. Okay, that is your negative staining. Now remember it as like this. There are two important negative stains. One is called as Indian ink stain, Indian ink, and another important is nigrosin stain. So everywhere you have N for negative, N for Indian, N for nigrosin. Okay, Indian ink stain, nigrosin stain. So if you are Indians, then use this stain because this is the cheap, this is very cheap and best. But costly and best is nigrosin. So the best stain to use nigrosin, but if you are budget friendly, then you can use Indian ink stain. Clear, clear, clear. Yeah. Now I'm going to show you some videos. Okay. What are those videos are? Before I show you those videos, let me write down these things first of all. Okay. So first important thing is that regarding the MCQ part, we are done with the staining, guys. We are done with the staining. We will also discuss culture methods. But before that, look here. We will discuss the motility of the bacteria. There are different types of motilities, guys. For example, there is a motility called as tumbling motility. Tumbling motility, right? T looks like L. L means listeria. So listeria performs tumbling motility. Listeria performs tumbling motility. Next, we have got darting motility. We have got darting motility. Dar stands for darling. Whenever your darling is beside you, you will have very good negative vibes, right? So vibes, V for vibrio. V for vibrio. So darling, vibrio. Darling vibes, darting motility is done by vibrio bacteria. Right? Now, another important thing. Swarming motility. Swarming motility. See guys, whenever you go for a swimming, right? Whenever you learn to swim. So first important thing is that you take training near a pro swimmer, right? An expert in swimming. He is also called as a pro swimmer. So whenever you're swarming or whenever you're swimming, you go to a pro swimmer, that is called Proteus. That is called Proteus. Swimming Proteus. Clear? Clear, 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 clear. Priyanka, thank you so much. Right. Next important thing is that there is also called as falling leaf motility. Falling leaf. Falling leaf. When you fall down, right? When you fall down from 10th floor, you're Gaya. Gaya means you're dead, right? So falling leaf, Gaya, that is Gaya dia. Falling leaf, Gaya, Gaya dia. Clear? And the last one will be corkscrew. Corkscrew motility. What does car stand for? Car. Car means vehicle. In the car, where do you go? You go for a trip. You go for what? Trip. Trip stands for treponema. Trip stands for treponema. So can you teach this conceptually? Now, how can I teach this conceptually? If a patient is walking, how can I teach conceptually that a patient is walking? These things have to be remembered. These things have to be remembered. Okay. The last one is lashing motility. Lashing means, for example, you go to a farm, you go to the village. What do? They take a whip and they lash the buffaloes. They lash what? Buffaloes. One of the buffalo name is called Borrelia. Okay, they lash Borrelia. So guys, these are the important things which you have to know, which you have to remember. Okay, which you have to remember. Okay, so let me show you the videos also. Let me show you the videos also. Look here. Right. What is this kind of motility which you can see here? It is falling like a leaf, right? This is called what type of motility? Falling leaf motility. Very good, very good, very good. falling leaf motility. Clear? Right? Very good, guys. Falling leaf motility. Next important thing is that look at this motility here. So this motility is in this way. You see, it is spirally rotating like a, what is this? Med booster. Very good. Swarming motility. No, it is not swarming. Roy. Very good. It is corkscrew motility. Roy, corkscrew motility. Okay, it is corkscrew motility. And let me also show you. I think I've shown you this. See, this type of motility, guys, this is called as, what is this motility? Anyone? I know it is not clear, but please try to relate it. Very good. This is called as swarming motility, right? This is called as swarming motility. Okay. And lashing motility also I will show you. Look at this. See, this kind of motility which you can see is called as lashing motility. You see there are lashes that is coming down like this. Lashing motility. Now this is corkscrew, that is different thing.
Showing the
so
flagellar
I hope you
tail.
understood these things. I hope you understood these things. Right? Now guys, all of you now pay attention. Now I'm going to discuss one of the very, very high yield topic. Very high yield topic with you. Okay, we are discussing very high yield topic, so please stay with me, because this will cover many MCQs. I'm telling you, this will cover many MCQs. Okay, so what is the topic? Gram positive bacteria. Within this gram positive bacteria, you classify this gram positive bacteria based upon catalase. I have already told you that we have got something called an enzyme called catalase. Angel, hello. Medbooster, hello, hi. I have already told you we have something called catalase, an enzyme called catalase. So catalase, some organisms are catalase positive. It means they release catalase enzyme. Some organisms are catalase negative, which means they don't release catalase enzyme. Organisms which are catalase positive are staphylococcal organisms. Organisms which are catalase negative are streptococcal organisms. Why are you telling this? Why am I telling this is that you will have number of staphylococcal organisms which you will study, Staph aureus and all. Whenever I discuss that topic, the first few points I will tell you is that this is a gram positive bacteria. This is a gram positive bacteria. Second thing, this is a catalase positive bacteria. Third thing I'm going to tell you right now. Okay, so classification is very important, guys. If you remember this table lifelong, I'm telling you, you won't even miss a single mark, because they're going to test you mainly on these characteristics. What is this bacteria, alpha hemolytic or beta, catalase positive or negative, gram positive or negative? So these are the things they'll test you. Okay. Regarding the streptococcus are further divided into based upon hemolysis. So what are the three types of hemolysis we discussed in the starting? Alpha hemolysis, beta hemolysis, gamma hemolysis. In alpha hemolysis also, you divide the bacteria based upon optochin sensitivity. Can anyone tell what is optochin, guys? Can anyone tell what is optochin? I am pretty sure that alpha hemolytic bacteria, all of you know, Viridans streptococci. There are three types of Viridans streptococci. I told you before itself. But right now I am forming another concept. What is optochin sensitivity? Optochin is an antibiotic. Roy, you are perfect. Optochin is an antibiotic. It is an antibiotic. Okay. Some organisms are optochin resistant. Some organisms are optochin sensitive. Some organisms are optochin resistant. Right? Kirby-Bauer method. Yes, very good. Now I know most of you might have known what is optochin, but some of you might have not known what is optochin sensitivity. Simple thing, guys. Simple thing. I take an agar plate, okay? I take an agar plate. I will divide the agar plate into two halves. One half, I will add, let us say, Streptococcus pneumonia bacteria. Okay, what I'm doing, I'm taking an agar plate, I'm dividing it into two halves. One half, I will add this Streptococcus pneumonia, S pneumoniae. Okay, on the other hand, I'm adding another bacteria. This is called as Streptococcus viridans. Listen carefully now. Don't write, listen carefully. Okay, what will I do is that I will take a tablet. Optochin is a tablet. What tablet? Antibiotic tablet. I will add that antibiotic tablet in the center here. I will add an antibiotic tablet in the center. Here also I will add. You know, antibiotic will kill the bacteria. If antibiotic is killing the bacteria, surrounding the tablet, whatever bacteria is there, all the bacteria is dead like this. All the bacteria surrounding the tablet is dead. But look in the Streptococcus viridans, all the bacteria surrounding the tablet is not dead. Why? Because Streptococcus viridans is optochin resistant. It can resist that antibiotic. It won't even listen to that antibiotic. So this is what I mean by optochin sensitive, optochin resistant. Okay. For your clear understanding, look at this slide here. Look at this slide. Now you will tell me, what is the center tablet here which I have added? The center tablet which I have added here, white color tablet, is optochin tablet. Okay, here also optochin tablet. Now, tell me, what is this and what is this? What is A and what is B? A is what? Streptococcus pneumoniae or Streptococcus viridans? Roy, optochin resistant is B. Yes, what is the example? A is pneumonia. Did everyone understood, guys? Did everyone understood? B first. Medical love, you are perfect. A is Streptococcus pneumoniae and this is Viridans streptococci. Very good. Very good. All of you. I hope all of you understood, right? All of you understood. Joshua, you're right. Joshua, I'm sorry, you're right. So now we shall be discussing about Streptococcus viridans, Streptococcus pneumonia. In the next class we shall discuss, but this class Streptococcus viridans. Okay. Now look the important things here. Streptococcus viridans. Sir, where are the MCQs? Every point which I have told you is an MCQ. Every point here. So this will be an MCQ. This can be asked as an MCQ. You know, even this can be asked as an MCQ. Even this also can be asked as an MCQ. Everything can be an MCQ, guys. Okay. So that is the reason why I'm not lagging it. I'm teaching clearly because I don't want you to mug it up. Okay. Right. Now you will tell me, Roy, I don't want you to answer. Medico Love, I don't want you to answer. Dr. Benedict, I don't want you to answer. But the remaining students, please answer me. Streptococcus viridans is gram positive or negative? I'm going to write 3 points here. Streptococcus viridans is gram positive or negative? Yeah. The remaining in the chart. Prajna, excellent Prajna. Pujita, Saumyadeep, Hasnain, Suchit, very good. This is a gram positive. Now tell me, it performs what kind of hemolysis? What kind of hemolysis? Prajna Barsa, perfect. It performs alpha hemolysis. Very good. Alpha hemolysis. Next, tell me, is it optogen resistant or sensitive? Obviously, I don't want to lag here. It is optogen resistant. So if you remember that table, guys, right? I mean, this is not the table. I mean, this is not the end of the table. As I will be discussing the topics, I will draw this table again and again and again. Every topic I discuss, I'll draw this table so that it would be very dead easy for you to answer these questions. Okay? It will be very easy. So what did I tell you? Viridans is of three types. They might, that is mitis, they might mute the songs, that is mutus, and songs, that is sanguis, right? S mitis, S mutus, S sanguis. Now, the first important question which they ask you is, what is the reservoir? Reservoir in the sense, wherever, what is a habitat, what is a normal flora? So all these are the normal flora of your oropharynx. Okay, so they are the reservoir, the normal flora of what? The normal flora of your oropharynx. Not only oropharynx, within the mouth you have got a beautiful teeth, and above the teeth you have got gingiva. So all these three together are the normal flora, where these bacteria, they live there very peacefully. Now let us enter into the pathogenesis. How am I going to get the infections that are caused by this bacteria? I hope all of you are maintaining a separate book for microbiology, right? What is the pathogenesis? There are different types of diseases that are caused by this. For example, guys, see here. All of you, don't write. I have these three bacteria in my mouth. Okay. I did not brush today. I mean, I brushed it, but let us say I did not brush today. Tomorrow also I won't brush. Day after tomorrow also I won't brush. For continuously for 1 week I won't brush. And suddenly I remember of my oral hygiene, and early morning I get up and I start brushing so vigorously. When I start brushing, don't you think there will be damage? Don't you think there will be some kind of injury to my gums? There will be injury. There will be blood oozing out of my gums. So when there will be blood oozing out of my gums, that is the time for this bacteria to enter into the circulation and cause damage to your teeth, damage to your gingiva. But this is temporary. This is temporary. That is the reason why the first disease or the first condition which we get is transient. Transient means temporary. Transient viridans streptococcal bacteremia. Okay, so this happens by eating, by toothbrushing, by flossing. Sudesh Berry, I love you too, right? But no, not that type of love. I'm sorry. Right. So right. So but anyways, transient bacteria, right? By eating, by flossing, right? By toothbrushing, you will get this transient bacteria. Second important thing. Second important thing. Can anyone tell me what problem in the teeth can lead to the development of the bacteria, the increase in the number of bacteria? Anyone? Yeah? Anyone heard of dental caries? Excellent, Roy. Perfect. Dental caries. Dental caries. I mean, not caring your teeth. Dental caries means there are, literally speaking, there are holes within the teeth. Okay. There is damage to the dentin. Okay. Now, how dental caries occur is that if you are not brushing daily, if you are not maintaining your oral hygiene, if you are eating all the waste material, whatever you are not supposed to eat, right? What will happen is that this S mutans and S mitis, S mutants and S mitis, both of them, they have a capability to release acid. They produce acid, and this acid will damage your dentin. This acid will damage your enamel on the tooth. All of you know, right? Enamel on the tooth. And what will happen? That would lead to dental caries. That would lead to dental caries. Okay. Another way, and another way that leads to dental caries is that what is another way that leads to dental caries, which is very, very famous in India? Yeah. You take this, keep it inside, and what is this called? This is called as gutka, right? So not alpha, it is gutka, right? So Indians, most of the Indians are gutka chewers, right? So whenever you chew gutka, then what will happen? The dental caries will happen. Okay, fine. So dental caries will happen. So doctor, I will remove my teeth. Then what is the problem? What is the problem is that, look here now. All of you concentrate now. I'm entering into the topic. Right. Let us say, these are your beautiful teeth like this, right? Above the teeth, what do you have, guys? I have these things called gingiva. Above the teeth, I have got gingiva. Whenever, whenever, and by the way, let us say these are the microcapillaries, the blood vessels. Okay, these are the blood vessels like this, gingival blood vessels. All of them, they join to form a larger blood vessel, and this goes down all the way like this, all the way like this, and it supplies to your heart. Now look what is going to happen. Whenever there is any kind of dental procedures, and by the way, remember this MCQ, guys. MCQ, this is an MCQ, that in case of dental caries, which concentration of bacteria or which bacteria are responsible for dental caries? Mutis and mitis, not sanguinis. Sanguinis is responsible for something dangerous. Look here. Whenever there is any kind of dental procedures, what will happen is that by chance, the gum might be injured, the teeth might be injured, the gaps between the teeth might be injured, so there will be bleeding coming out. So that is the moment when during the dental procedure, that is the moment when this sanguineous bacteria, whatever is there, it enters into your circulation like this. You see, from here the bacteria has entered into the circulation. Before entering into the circulation, it will damage all the teeth, it will damage all the gums, right? So if it is damaging all the gums, you call this condition as gingivitis. And what will happen is that this bacteria moves all the way down, all the way down, all the way down, all the way down, and finally this bacteria will come to the heart. What will happen when it will come to the heart? Look here. This is the bacteria. Surrounding this bacteria, another MCQ here. Surrounding the bacteria, you have got a very special layer. Now, this special layer surrounding the bacteria is called as dextran. What is this? It is called dextran. Remember it, very, very important MCQ. It is with the help of this dextran, the bacteria very closely and very strongly, it attaches to the heart. It is with the help of the dextran, the bacteria very closely and very strongly attaches to the heart. And you know, once the bacteria attaches to the heart, what will happen? The outer layer is gone first. Outer layer is damaged, and that condition you call it as pericarditis. After that, the bacteria will penetrate a bit deeper inside, and that will cause myocarditis. And further, it will enter into the endocardium and cause endocarditis. Whenever patient will have endocarditis, I will put a stethoscope on his chest and look and listen to a new murmur. Okay, if you listen to a new murmur on examination, that is mostly endocarditis. So what are the MCQs that are asked from this entire topic? See here. These are the MCQs which are asked. First MCQ. Second MCQ. Second MCQ is this one. Dental caries is caused by. Third MCQ is because of dental procedures, sanguinus is causing endocarditis. Right? Dental procedures leading to endocarditis is Streptococcus sanguinus. This is also an MCQ. Okay. Now let us do this question here. All of you might have known this by now. A 56-year-old patient reports a history of 56-year-old patient reports a history of recurrent dental caries. Recurrent dental caries. The bacteria causing this patient's dental problems most likely exhibits which of the following properties? Which of the following properties, guys? Is it inhibition on the growth on optogens? Is it optogen resistant or sensitive? Yeah. Resistant. But here it is telling inhibition of growth. It means it is sensitive. But this is not our option. It is a gram negative diplococci. Is it a gram negative diplococci? Okay. Tell me, is it a gram negative diplococci? No, it is not a gram negative diplococci. Presence of catalase enzyme. Does this has catalase enzyme? Yeah. Is it catalase positive or negative? Let me go back. I have already discussed this, right? Look at this. It is catalase positive or negative? Benedict, look at this. It is catalase negative, not positive. Streptococcus comes under catalase negative. Clear? Everyone? Yeah, negative. It is catalase negative. Soluble in bile, we did not discuss about solubility, so leave it. Catalase, presence of catalase enzyme, no. So, very good, med booster. Your answer is perfect. Production of dextrans from glucose. You might tell, you told me dextran, then what is glucose doing here? Simple thing, bacteria has glucose will release dextran, and dextran will go outside the bacteria and settle outside the bacteria. That's it. Yeah, everyone understood. So the answer is 3. Answer is 3, right? Very good, guys. Excellent, excellent, and excellent. You know, we have covered a lot and lot of MCQs, lots and lots of MCQs, guys. You see, this is dental caries. This is a picture of dental caries. Now let me show you some beautiful teeth here. Now this is beautiful teeth. You know, most of them tell that the dental studies are more famous or more good in Australia. But once the Australian doctors, when they come to India, they'll be shocked to study the new anatomy of the teeth here. This is the teeth of a gutka chewer. How beautifully he has colored his teeth. Yeah. So this is all the dental care is whatever is having. Okay. Gingivitis. Dr. Benedict, you are right. Gingivitis. And MedBooster is telling that dextran is non-soluble. Yes, that is the reason why dextran is very happily sticking to it, because no one can destroy it. Bolo Juban Kesari, yes. Yes. Yeah, you're right. Colorful, yes. So some of the foreign students in this, they might be really surprised, but this is usual color which we daily see, you know. So this is the endocarditis component, guys. If you can see here, all this yellow color plaques, these are not yellow color plaques, these are the vegetations. The bacteria has grown there. These are the vegetations in the endocardium. See, this is a classic picture of endocarditis. Dr. Benedict, you're right. Now uh, Let us do a small test, guys. Let us do a small test here. Now tell me, what kind of bacteria is this? Is this a alpha hemolytic or beta hemolytic or gamma hemolytic? Yeah. Yes, I want your answers. I'm waiting. This is a small test for you. What kind of bacteria is this? Not beta. There is no golden zone of hemolysis. Joshu Gawai, very good. Pujita A, very good. Kyle, Dr. Benedict, look here. This is a bacteria. Surrounding the bacteria, is there any zone of hemolysis? No zone. Surrounding the bacteria is completely red. It means no RBC has been hemolyzed. So this is a gamma hemolytic bacteria. This is a gamma hemolytic bacteria. What is this? It is a gamma hemolytic bacteria. Whereas this is a beta hemolytic bacteria. Compare these two pictures, Benedict. Look at this and look at this. There is zone of hemolysis here. There is no zone of hemolysis here. Right? And you will tell me what kind of hemolysis is this? Observe carefully. What, if you want, I'll enlarge the picture little bit more. What kind of hemolysis is this? Very good. Partial hemolysis. This is called as partial hemolysis or green hemolysis. Partial or green hemolysis. Okay. Very good. You're right. Right, guys. Right. Fine. So tomorrow's class, we shall be discussing very different kinds of bacteria, and we shall also be discussing the culture methods. Okay. So we shall be completely focusing tomorrow on the separate aspects. Okay. Separate aspects. So guys, did you understand whatever I have taught you today? Is everything clear today? Is everything clear? Or is there any difficulty that you didn't understand? Tomorrow at the same time. Tomorrow at same time, 6 o'clock. Everyone, I need feedback from everyone, guys. Should I change anything? Should I change any of my way of teaching? Should I improve anything in the next class? Your feedback is very valuable for me. Right. So I hope you understood the session. Uh, for quick review, yes, we are also planning quick review for all the subjects. Right. They will start from November 10th. From November 10th, we will have quick review of anatomy. We will have quick review of all the subjects. Okay. So we will have a long session of about 7 to 8 hours, and we shall discuss everything. Okay. So, okay, quick review. Quick review at the end. There is nothing here to discuss, guys. I told pigment producing bacteria. I told you four types of pigment producing bacteria. Hemolysis, I told you alpha, beta, gamma. I also made you to practice this hemolysis. You are perfect with that, right? Examples you should study. Bacterial virulence factors. These are the three virulence factors. Okay. And finally, gram staining methods, three staining, simple, differential, special. Simple means I use one color, differential means many colors, special means specially. Okay. So in case of differential, Gram staining, Zeal nasal staining, are also called Zn staining, right? In special staining, there are saint. A stands for Albert, I stands for impregnation, N stands for negative staining. Impregnation, I told you, silver salt you add, the flagella will swell, you can study it. Okay, negative staining, bacteria will stain, background will stain, capsule will not stain. Okay, capsule will not stain. So these are some of the things, guys. So once, anyways, we shall post these notes in the group. Okay, in the Telegram group, we are posting these notes. So yeah, so you can have the notes over there, and it will be pretty easy and pretty simple, guys. Tomorrow, guys, I'm gonna teach you very solid, important topics, and I'm gonna teach you very interesting topics, guys. So tomorrow, I want all of you, right, to be on time as you are today. So, but anyways, thank you so much, guys. Thank you so much for supporting me. Love you all. Goodbye.